Hello and welcome dear students, professionals and learners to the another topic on impurities. So in this topic we will discuss impurities in new drug products, their acceptance criteria and qualification requirements. The ICH guideline Q3B is referred for this video and it will cover the introduction to the organic impurities, then what are the types of organic impurities and how the acceptance criteria is based on the impurities, their limits, and the qualification requirements. So, impurities. First of all, we have to understand what are the impurities. So, these are the impurities are other than the drug substances or ingredients that arise out of the synthesis or unwanted chemicals that remain with the active pharmaceutical ingredients, that is APIs, or developed during the drug product formulation. So impurity is any component of the new drug substance that is not the chemical entity defined as the new drug substance. So these are the unwanted chemical compounds present into the API or into the drug product, and these may come from synthesis of the APIs. These may get arise from the process. These may get formed as degradants in the formulation. So mainly the organic impurities are there, inorganic impurities are there, and the residual solvents are there. So all these are treated as impurities. So organic impurities, then inorganic impurities. So inorganic impurities are elemental impurities, and the nitrosamine impurities are also there. Then some of the genotoxic impurities are also there. The nitrosamines and genotoxic impurities are considered in the organic impurities. Then residual solvents. So residual solvents may come from the process, or these may come from the manufacturing of the formulation. For example, if acetone is used in the manufacturing, so this residual solvent acetone will come from that manufacturing stage. Then considering the organic impurities, this may be the synthesis process, or maybe during the storage of the substances. So from these may come from synthesis process, or maybe formed during the storage of the drug substances or drug products. So process impurities arise from the processes like intermediates are there, starting materials are there, catalysts are there, ligands are there, and intermediates are there. Then degradation products arise from the storage. If the API get degraded, that degradation products will be formed, and these arise from storage, synthesis process, and formulation of the product. So these are also formed in the formulation. The degradation products may be formed due to the excipient interaction or due to the aging of the formulation. Then organic impurities, as we have discussed, these are synthesis, process impurities, byproducts, intermediates, reagents, ligands, catalysts, or may be formed during storage of the drug substances and the drug products. Process impurities arise from the process like intermediates and starting materials. Degradation products arise from storage, synthesis process, and formulation of the drug product. Organic impurities are controlled with the specific acceptance criteria based on the threshold criteria as per the ICH guidelines. So, how these impurities are controlled and what are the guidelines? So, specific guidelines are available for the specific type of impurities, and ICH Q3 guideline deals with the organic impurities mainly, whether it may be in the drug substance or drug product. So, these organic impurities are controlled with specific acceptance criteria. Why? Because these impurities, if present in the drug substance or drug product above certain level, may prove to be toxic in nature for the human consumption. So there are some definitions which are required to be understood before going ahead for the impurity calculation, impurity acceptance criteria and qualification. So impurity may be identified or unidentified. Identified impurity is the impurity for which the structural characterization has been achieved. That means it is a known impurity. Because its structure is known, so it is also called as known impurity. Then specified impurity. The specified impurity that is individually listed and limited with specific acceptance criteria in the new drug substance specification. So this is specified. We have to specify the impurity with particular limit and the acceptance criteria, and this is individually listed. For example, impurity A or impurity at RRT. So these are the specified impurities. So these specified impurity may be either identified or unidentified. If it is identified, it is known as known impurity. If it is unidentified, it is called as unknown impurity. But it is specified and it becomes specified unknown impurity. So unidentified impurity is an impurity for which a structural characterization has not been achieved, and that is defined solely by the qualitative analytical properties like chromatographic retention time. We call it as unidentified impurity on RRT basis. It is the specified impurity, but it is unidentified because its structure has not been characterized. That's why it is specified but unidentified. Then unspecified impurity, an impurity that is limited by general acceptance criteria but not individually listed in its own specific acceptance criteria in the new drug substance specification. It is also called as unknown impurity. This is unspecified. So if you see the types of impurity, the impurity may be known or unknown. So known impurity will have a specific name, and unknown impurity may be specified or unspecified. As per the guidelines, impurities are controlled by the specific threshold levels, and threshold is the level at which something starts. That means these are the limits or the level at which the impurities are required to be controlled. So three thresholds are given. First is reporting threshold, second is identification threshold, and third one is qualification threshold. You can remember this by taking the initial of these three thresholds as RIQ: reporting, identification, and qualification. Now if you have an impurity which is having a level which is below reporting threshold, so it is not required to be reported, and reporting means it will not become the part of impurity profile. Impurity profile is the list of all impurities, known, unknown, and specified, unspecified. All the impurities will have a list along with the total impurity. That is called as impurity profile. So if the impurity is having level below reporting threshold, it will not be reported. The limit above which the degradation product should be reported is called as reporting threshold. But once it crosses the reporting threshold, it will be reported. The reporting threshold is also called as the disregard threshold. Then identification, the limit above which the degradation product should be identified. Now we are dealing with the degradation product. The degradation product may be known or unknown. If the degradation product is unknown, then first it will go through the reporting threshold, then identification, and then qualification. If it is known, it will directly go to the qualification. So that we will discuss in detail in the next upcoming slides. Now identification threshold. If you have a impurity level in your product and it is above the reporting threshold, so you will report it and you will give a level of identification. If it goes beyond the identification threshold, it will go to qualification level. Now the qualification threshold is the limit above which a degradation product should be qualified, and qualification means acquiring the safety data for impurity. The example here I have given is the maximum daily dose is 25 mg. Consider a tablet or capsule with maximum daily dose of 25 mg. Its reporting threshold will be 0.1%, identification threshold will be 0.2%, and qualification threshold will be 0.5%. If the impurity is having a percentage of 0.05, it will be disregarded. If the impurity is having a level percentage of 0.15, then identification threshold limit will be given as 0.2%, and if it goes beyond 0.2, it will have a qualification threshold of 0.5%, and if the impurity goes beyond 0.5%, then qualification is required to be done. So always remember this RIQ, that is reporting, identification and qualification thresholds. Now reporting threshold, identification threshold and qualification thresholds we will see. See, reporting threshold, it is mentioned that maximum daily dose and the threshold. For the drug products with maximum daily dose of 1 gram or below, the threshold of reporting is 0.1%, and if it is above 1 gram, then the threshold of reporting will become 0.05%. So one of the simplest impurity calculation is for reporting thresholds, where you don't have actually to calculate. It is already given there. You just convert the maximum daily dose into percentage and you will get the threshold limits. Then coming to the identification thresholds, in this they have mentioned maximum daily dose and threshold is given for each category of the maximum daily dose. For the drug products with maximum daily dose below 1 mg, the threshold limit is 1% or 0.5 microgram TDI, which is lower. Then similarly, the limits are given into the qualification thresholds also. So TDI means total daily intake, whichever is lower. So whenever you are calculating the impurity limits or you are doing impurity calculation, these small words have a great meaning. For example, identification threshold for 1 mg, for less than 1 mg, it is 1% or 5 microgram, whichever is lower. So always you will find this terminology as whichever is lower. Means the limit for your maximum daily dose, after calculation you get, you have to consider the lowest limit. These limits and the calculation we will start now. So reporting threshold is one of the simplest that I have mentioned earlier. Now coming to the impurity acceptance criteria. This is very important topic and it will need your 100% attention so that you can decode the guideline and have a complete clarity of the topic. See, if the degradation product is present into the formulation and you know that the degradation product is formed due to the process, manufacturing or due to the aging of the formulation, it may also be formed due to interaction with the excipients or due to the storage temperatures. So there is a degradation of the product or API and degradation product is formed. So we have to check whether that degradation product is greater than the identification threshold. If it is more than the identification threshold or not. So if it is not more than identification threshold, then give a limit of unknown impurity, that is identification threshold limit, and there will be no further action because you have controlled the degradation product by identification threshold. Now if degradation product is greater than the identification threshold, then we have to identify the structure and we have to check whether that structure is having any relevant risk, that is toxicity to the human beings. So the answer may be yes or no. So the answer, if it is yes, then it is required to reduce to the safe level. So once again we will see, is degradation product is greater than the identification threshold? The answer is yes. Structure is identified? Yes, we have the structure. Now, is there any known human relevant risk for this structure? Yes, the risk is there. Then reduce to the safe level. So this is example 2. Now, is degradation product greater than the identification threshold? Yes. Structure is identified? Yes. Any human risk or any known human risk? Here it is no. In earlier example it was yes, so it was told to reduce to the safe level. Now it is not having the relevant risk. So once again we have to check, is it greater than the qualification threshold? It is no. Then give the limit of known impurity, that is qualification threshold, and no further action will be there. In simple meaning, we have to test whether the degradation product is above the identification level or above the qualification level. If it is above the qualification level, we have to qualify. Otherwise, the qualification threshold will be crossed. Then is the degradation greater than the identification threshold? Yes, it is above. Structure is identified? No, structure is not identified. Here structure identification is not there. So reduce to not more than the identification threshold. See, structure identification is nothing but making the impurity known. If impurity is made known, then we have to give a qualification threshold. And if your known impurity crosses the qualification threshold, that time qualification is required to be done. If structure is not identified, then reduce to not more than the identification threshold. And if that is not possible, then we have to qualify the impurity and give the limit as qualified. So qualification is the process of acquiring the biological safety. The process of acquiring and evaluating the data that establishes the biological safety of the individual degradation product or a given degradation profile at the level specified. So generally the impurity qualification involves the consideration for patient population and duration of use. So for qualification of the impurity, genotoxicity studies are done, and these are called as minimum screen for genotoxicity. Involves the point mutation study and chromosomal aberration study. Then general toxicity studies involve the general toxicity study with rodents with one species. Usually 14 to 90 days are sufficiently considered. Then other specific toxicity endpoints as appropriate for the impurity. So this is regarding the impurity acceptance criteria and qualification requirements for the impurity. The guideline mainly referred for this video is Q3B R2. that is ICH guideline on impurities in new drug products. So I hope you might have got good understanding of the impurity acceptance criteria and qualification requirements. So please understand that these topics are very important wherever you are working in the pharmaceutical industry, whether you are in QC, QA, regulatory affairs or formulation R&D. So the understanding of impurity is much required, and impurity qualification and impurity acceptance criteria is the favorite question for the many of the interviewers. Thanks for watching the video. Please do like, share and subscribe to Pharma Learning Ed channel.